come to where the microtubule are found. So the microtubules are found in the cilia, flagella as well as in the centrioles. So first come to the cilia and the flagella. Cilia, another is the flagella. So what is the cilia and what is the flagella? They are the locomotory organ. What is the locomotory organ? That means it helps in the locomotion of the organism. For example, the cilia in the paramecium. In the paramecium, the cilia, this like structure is present and they are called as the cilia. For example, the euglena. In the euglena, a tail-like structure is present and that is called as the flagella. So that means the cilia, it is the singular term is called as the cilium. That means one cilia is called as the cilium, while one flagella is called as the flagellum. Okay. So now the number of the cilia are always high, but the number of the flagella maximum one or the two, or mostly the one flagella is present, or the single flagella is present, or the single flagellum is present, or maximum two flagella are present. For example, in the euglena, the flagella is present. In the paramecium, that means it looks like the slipper. So that's why the paramecium is also called as the slipper-like animalcule. So in the paramecium, the cilium is present, and in the euglena, the flagella is present. So now, in the cilia, or the animals which are the cilia, so that means all the cilia must have moved at a time, so that it can move to the forward or to the backward. But in the flagella, because of the presence of single or maximum to two, that means it is not coordinately manner. That means it can move independently, while the cilia must have coordinately moved to each other, so that organism can move to forward either or backward. So now the cilia, it runs like a running manner, or it moves like a running manner. Running manner means all should have, or all runs like this, but in the flagella it presents, or it moves like wave-like manner. So this is the main difference between the cilia and the flagella. So now look at the structure of the cilia and the flagella, or the microtubule which is present in the cilia and the flagella. And but before going to that, for example, this is the paramecium. Okay. So the cilia structure, it looks like this. So that means this is the whole cilia structure. So now some part of the cilia is protruded in the cytoplasm, and some are present in the outside. So the outside part of the cilia is called as the axoneme. This is very important, axoneme. While the part which is present inside the cytoplasm or inside the body, that is called as the basal body. Again, it is the same for the flagella. That means flagella also, it has the same structure. That means the outside flagella structure is called as the axoneme, while the structure which is present inside the body or which is present in the cytoplasm is called as the basal body. So this is the main difference between the cilia and the flagella structure. That means the cilia and the flagella structure, they have two different structure. One is called the axoneme, another is the basal body. And the structure of the axoneme and the basal body is that obviously the microtubules are present inside the axoneme or inside also in the basal body, but the structure are very different. So now look at the structure. So now first we should look about the axoneme structure. So now I am telling about the exonym structure. That means the structure which is present outside the body is called as the exonym structure. So now in the exonym structure, the structure looks like this. So that means here they are called as the peripheral ring. So what are these structure? For example, as we have discussed earlier, this structure. So here, for example, two ring like structure is present. So that means one structure is like that. That means 13 protofilaments are present, and to that again one, for example, this is the one tube, and to this another tube are joined. Same like structure. So that means if we take the cross section, it looks like this. That means one, another is the another structure. So this is the main structure of like this. So this is called as the peripheral ring. So in the exonym, the outside ring are called as the peripheral ring, and they have 9 peripheral rings. This is the first, this is the second, this is the third, fourth, fifth, sixth, seventh, eighth, and the ninth. So that means 9 protofilament like structure, the 9 microtubule structure is present in the peripheral, and in the central region, this is the central region, this is the central region, or it is called as the central ring, and in the central region 2 structure is present, one, another is a 2, but here they are present like in the diploid structure or the doublet structure. This is the doublet structure. So the exonym structure, as we, or if we look the structure, the exonym structure, it is designated as 9+2 arrangement. What is the 9+2 arrangement? So 9 means in the peripheral structure, 9 structure or the 9 microfilament structure is present, and which are the doublet. But here the 2, that means the single structure, and that is present in the twice. This is the one single structure, this is the another single structure. That means 2 single, that means it is the 2 singlet. So that means 9 doublet and 2 singlet, and the 9 doublet are present in the periphery region, and the 2 singlet structure are present in the central region, and the all overly it is called as the 9+2 arrangement, and it looks like this. So now this is the central region, and in the central region the central link is joined to the peripheral link by this structure, and this structure is called as the radial spoke. So that means the radial spoke, it joins to the peripheral link to the central link, while the peripheral links, they are again joined by the connectors. These are by connectors. So these connectors are called as the nexin. So these are the connectors, or they connect the peripheral link structure. So now to this, or to this peripheral link structure, a structure like this structure is present. Here the 3 dot, if we series, so this 3 dot structure, this is main important structure, that is called as the dynein arm. So this dynein arm, it helps in the, or it releases the energy for the movement of the, for the movement of the organism. So that means for movement, some energy is required, and this energy is released by this dynein structure. So this is the all about the exonym structure. So now come to another structure, that is called as the basal body. So that means, for example, this is the paramecium, this is the cilia structure, and this inside the body or in the cytoplasm structure, the centrioles are present. They are called as the centrioles. So now in the basal body, the centrioles forms the cilia and the flagella, and the process is called as the ciliogenesis. So what is the ciliogenesis? So that means inside of the body, or the inside, not our body, that means inside the for example the paramecium or the euglena. So this structure, so here the centrioles forms the cilia and the flagella, and the process is called as the ciliogenesis. So during the ciliogenesis, the centrioles are called as the basal body. So this is the again another structure of the cilia and the flagella, which is present inside the body or inside the cytoplasm. So now the structure of the basal body is different. Look at the structure, and this is same as present in the centrioles.